Luck, y'all.
Good morning, everyone. Welcome back. We left off still talking about plant growth and development. We were talking about the hormones, plant growth hormones, and we had just talked about auxins and cytokinins. So we'll resume there, and then we'll move into the next chapter on plant chemistry. Before we get started, I just want to throw out there that the midterm exam is due November 1st. That's also when your crop project is due, at least the first half of it. So just don't let that stuff go by the wayside. Also, the quiz for this week is, or for this chapter is due a week from today. So we'll finish that up. For the midterm, it's not up right now, but it will be up by the end of the day. I just finished the review this morning, and I just have to fix a few little errors on that test, and then it'll be ready. But you've got some time to do that. It is through Canvas, and it does have that Respondus lockdown browser where it's only the time limit. So it's before taking the exam, because I'm going to go everything that we've covered up until now and go through any of those questions that there was a lot of contention or debate about, and especially the confusing ones. Some of them I have changed a little bit, but a lot of them are coming kind of directly from those quizzes. It's just measuring how much of that you actually absorb. Development. Again, the cytokinin. And in this image, we're looking at a plant known as tissue culture, which we'll talk about in more detail when we get to propagating plants. We'll take just a few cells of a plant work and then photosynthesize, because you're giving a that it grows and trigger the different changes in the plant seen in this image with tobacco. Just go from just this basically a piece to get it to grow a vegetable fruit, get the correct different plant growth hormones. So develop new plants. Neat that we can do that. And thing for tissue culture is that you can produce a ton more plants. They're all clones, but a ton more from just one piece of a plant than having to just take a few cuttings from a plant, for instance. We can preserve those much longer. So when you about like a seed banks, stuff like that, a lot of times they have tissue culture vial and test tubes pretty much, so that they can later time activity and DNA. The next, ethylene, is a different plant. We talked a little bit last week about how it can affect the gas. It's easy throughout the plant to CO2. So synthetic ethylene as well in a liquid form. Again, the ethylene is in the there are a couple also get it to initiate flight. It has to also be along with the photo environment. You can also change the expression of flower. You can use that to person. As with, it's kind of the same process in things like get them to change applying ethylene, drop their fruit, and so it came by causing a more quick the abscission of leaves. Don't store those. You got your apples near your onions for your onions to sprout. You can also call the flower bud abortion of bulbs during shipment. Another thing to look at for ethylene is that gas or oil fired heaters can also generate ethylene adequately. So, all right, our is abscissa, which the major thing with ABA is that it's of the growth and development, and also kind of in the abscise. And it's also a stress hormone. So respond too little of the plant as of now, and plants deactivate it. So here's just a couple of other as the major natural hormones, but they're just still the brasinoids are steroids, and actually have been doing it for a long time. They use that chemistry to extract that, to use that for and that is just one example. Um, Salicylic acid, which is kind of the same as same chemical, also important for the plants, and are and basically same deal um, against a unique one that and involved in the so these are, in fact, most of them are unique to a family or just a genus. Percentilides are in the brassicas. Salicylic acid is in jasmine. So not in all plants, but plant scientists are trying to figure out ways to use these to basically have almost like as a pesticide in a way, except kind of the opposite. It's not killing something, it's better preparing the plant for certain attacks. Then we also have some synthetic just growth retardants, and basically kind of that same principle of synthetically applying them, mostly in the ornamental industry, just to get kind of compact dwarf type plants that are more aesthetically pleasing. And some of these, we can also get more attractive foliage and more flowers than in plants that are untreated. So not that you have to memorize any of these, but here are just some of those synthetic growth retardants out there. And so we've got the kind of chemical name, well, actually the scientific chemical name here, the kind of more generic chemical name, and then over here we've got the product name. So the only one that I've really used is paclobutrazol, Bonzi, which is just used to basically keep plants bushy and compact. And I think some of these are probably also like B9 I've seen on the market. I've never used it myself, but really just in the ornamental industry do we see these. All right, so that covers our plant hormones. Anybody have any questions there on chapter 7, plant growth and development? Well, that's a good segue into chapter 8, which is getting industry. And plant chemistry and metabolism. It's important to know the processes behind it, why we need particular elements and nutrients and what they do. But after in the book, it's dense. I just don't believe that you all need to memorize any of these, chemicals. We'll get more into this plant chemistry and metabolism when we get into chapter 11 in photosynthesis and respiration. But again, in both of these chapters, I reach the level of plant science. A whole lot from the chapters here. Trying to get the most crucial, starting off with just concept art, just like any other chemical in the plant. It's a molecule that is involved in the makeup of the plant itself. And it's involved in part of the structures of the plant, the tissues that make up the stem and leaves. Basically, we're really all thinking of metabolism in plants, just like metabolism in people. Metabolites are what the plant is making in the leaves and then using basically to do everything else, to compose either plant nutrients or taken up for them to function. So this all begins with the creation of occurring in the leaves, the little solar panels there. So we got side of the chlorophyll, we have the creation of carbon. Plants are in on kind of concept molecules, and for energy, movement of energy, go through every. So let's just break it. The most simple form of carbohydrate, coming from the word itself. Hydrate being water, of course, H2O and carbon. So Carbon, hydrogen, and oxygen are kind of what we would refer to as organic chemical. They compose life, basically, the dry matter of plant water out of it. What is carbon, hydrogen, and oxygen? Carbon, hydrogen, and oxygen from it from the plants actually absorbing carbon dioxide, get from water taking up, or sometimes basic matter in the soil was once plants. So compose generally basic formula with a water molecule, basically. So 2 hydrogen atoms and 1 oxygen. Structural groups are very important. It's are classified. It's basically bonding of the carbon. Just between 2 structural groups, an aldehyde group. I'm not going to get into much more other than just the fact that of bonded together to each other to make the difference. So we've got oligo and polysaccharide, different arrangements, numbers of how these disaccharides are so linked together. Excuse me. Common and important because it's structure that is set is glucose, and it is used pretty much and we're looking at it in equilibrium plant forms. Carbon atoms in the molecule are and basically help to keep track of the different and these vertical structures groups, so the hydroxyl hydroxide to it, and where it's attached, glucose. At its very basic form, O6, but then it's ways to give different in the plant. So carbohydrates, it determines that ring structure are the actual forms that are involved in the plant's metabolism itself. And if we change that structure, then we have a different compound with different chemical properties created. We get down to cellular DNA and RNA, and I'm also the plant structure from the different chemical structure of glucose itself. All right, so what I think is more important than the different structures of glucose is how they actually work and what they actually do. So here we have the general structure of and is important because it parts of plant to differentiate it between have one similar to make the structure of cellulose. So the only difference in the structure itself of this hydroxyl carbon attachment of the glucose. So just one slight difference in the chemical makeup of that carbohydrate of glucose changes it from a starch to, and even though it's one simple chemical nature of the first, starch is slightly and can be broken to the simple glucose for our nutrition. We down into the simple carbohydrate, which we make other things, basically everything. So it's driving the plant, driving our metabolism. Cellulose, on the other, insoluble in water, unless you do things to make it soluble, and it's assembled into fibril, like adds the rigidity required for cell wall. So for kind of more for energy, cellulose is more as a structural component. We cannot digest cellulose very easily. Organisms cannot digest cellulose very easily. Cellulose becomes very important for organic matter, but just for the body. So even though we have fairly similar structure of the 2 molecules, is how they are used in the plant and very different. Common carbohydrate is sucrose, and it's used in carbon transport. The disaccharide molecule create sucrose. So it's the same chemical structure, and again, it's an important. Sucrose and fructose are used differently in plant, and carbohydrate are plant chemicals that is important to look at is, and these are compounds that are derived from molecule, and these are important both in the cell membrane structure and also very important for our metabolism and a lipid in order to have a functioning metabolism. Lipids are hydrophobic, so they are not soluble in water, wide variety of fatty acids. But here again, arranged in all kinds of different ways in the plant or in the animal, whatever it is. How we classify them as the subscript N of it, that just means that this group of CH2 here, and depending on how many times it is present in that chain, we get the different fatty acids. So lauric acid has 10, palmitic acid has 14, stearic acid has 16. So, and there are many more of these fatty acids out there, but just to give you an example of how that structure is. These fatty acids can either be saturated, or we say that we want to get more, unsaturated fats, because of it's really just looking at how these fatty acids are bonded. So oleic acid is an example of an unsaturated. So this is, I know it looks kind of painful, simple form, even though it looks very complex. But again, it's just this. So it's a linkage of fatty acids to a glycerol. So we have materials such as fats or waxes and oils in the lipids. And just it goes back to the organic elements, just a different arrangement for it to give a different function in the plant and then in us as well. One of the major functions of these in plants are as membranes. They're attached to a important as to how it acts in to give this structure, coupled with the different structure of the different fatty acids. These membranes have different functions, most cells of the plants, which right now the ability of the plant to make energy. Two different chemical surfaces, one that is hydrophobic and repels water, and take in different chemicals that might be transported. So here's just a look at a kind of lipid arranged in a double layer due to the mutation chains. So this arrangement gives the basics exchanging kind of proteins here. So we've got extrinsic out and in and out of the cell itself. And these proteins are carriers for different plant nutrients. Cool temperatures as well. Think about their maybe not evolutionarily the best thing, more unsaturated fatty acids in their membrane. They will start to basically the chemicals and at or below 55 degrees, which is why the tropical and subtropical plants. So fats are important in cooler temperatures. The difference in saturated nutrition as well. So, and the types of oils that they provide. Just thinking about going back to our first lab and we, modified synthetic foods, but highly processed foods, fats in them, because they it's one of the big ones. So anyway, you get saturated fats from unsaturated fats are good, saturated fats are bad. They just let's talk about the sterols, which is not in plants. We don't have cholesterol. Those are inserted into a membrane of fatty acids. The mineral content of some plant membranes can reach 50%, so half related to cholesterol, which is the main, very similar, except in cytosterol. All right, so hopefully that explains the list. Next, let's look at the proteins molecules than our carbohydrates or lipids. And they have a whole bunch of different functions as well. They can serve as a storage for nitrogen, chemical reactions. Proteins are extremely important in everything that we do as well. So we make protein. become much more complex carbohydrate, but adding nitrogen into that formula, common structure. So ammonium is combined with a carbon compound, types of amino acids that are out there. And in this case, this is a process called with looking into how these are. Ammonium is where we're getting that nitrogen group in there, kind of taken into this chemical process where proteins, and they are arranged in a very 8 of those amino acids are here: methionine, valine, leucine, isoleucine, about lysine and methionine, kind of early on in the agriculture, kind of an grain and legume combination. And remember, the methionine together, we get all 8, but together you get this complete set of the 8 essential amino acids. There are many more, and your health. The 8 essential amino acids are actually creating amino acids. They are also the 8 kind of most common. Those 8 cannot be synthesized by animal protein, either directly from eating your kind of grain, those plant proteins. So, I'll just mention this to you all. Trying to figure out how you can, better human nutrition in order to break those proteins down into his concept. And there's been a fair amount correct, not really out there, of plants that had more of these free amino acids. His concept is that it takes along amino acids. There are other parts in a short term. So we're metabolizing the vitamins, say an hour for our bodies to break them down into the amino. Might be better for your nutrition. That's better. I think ideally we get a lot of these free. Our grain and legume combination is important, but we need. I'm not a nutritionist, so I'm just going to shut up now and cure animal science does.